All right, so at this point, we have completed our discussion on the anaerobic systems. So to summarize the two anaerobic systems, first, the phosphagen system. This again is the most rapid system in that it produces ATP at the fastest rate. And since this is an anaerobic system, it will kick in primarily during post-anaerobic threshold exercise, and its activity is highest during max intensity exercise, like all-out efforts such as sprinting. The phosphagen system utilizes phosphocreatine as a fuel source and transfers energy from it and uses it to rephosphorylate ADP to ATP through a single reaction catalyzed by creatine kinase. The next anaerobic system, again highly active during post-anaerobic threshold exercise, is fast glycolysis, or the lactic system. This system involves only the first portion of carbohydrate or glucose 6-phosphate oxidation, which is glycolysis. This is the first step of carbohydrate oxidation, which is anaerobic. It does not require oxygen. This means fast glycolysis is just partial oxidation of glucose 6-phosphate, ending in pyruvate converting to lactate. Remember, full oxidation involves pyruvate being further oxidized in the mitochondria via aerobic metabolism. So during post-anaerobic threshold exercise, an increasing amount of glucose 6-phosphate are being oxidized fully through glycolysis and into aerobic metabolism. But an increasing portion of the available glucose 6-phosphate undergo only partial oxidation via fast glycolysis, ending in lactate and providing just 2 to 3 ATP rapidly. Fast glycolysis activity increases right around the anaerobic threshold. So therefore, since lactate is the product of fast glycolysis, we can test the exercise intensity at which a subject reaches his or her anaerobic threshold by testing blood lactate. So during an exercise test of multiple stages of increasing intensity, we can test the blood lactate at each stage, and eventually at a certain stage, the blood lactate will begin to rise rapidly. This is indicating that the activity of the lactic system, fast glycolysis, has increased. And it also indicates the anaerobic threshold, since remember, the lactic system increases when you hit your anaerobic threshold intensity. This is why the anaerobic threshold is often referred to as the lactate threshold. So the exercise intensity at which your blood lactate rises rapidly is the anaerobic threshold intensity. All right, now let's move on to the muscle cell's main bioenergetic system, the aerobic system. This is again represented by the white area under the curve in this figure. The aerobic system is always active no matter what intensity of exercise and level of ATP demand by the muscle cell. So here are some general characteristics of the aerobic system. Aerobic ATP production involves the full breakdown or oxidation of carbohydrate or fat with the help of oxygen. So again, the carbohydrate fuel molecule is glucose 6-phosphate, which can be derived from glucose that is taken up from the blood or glycogen that is stored in the muscle cell. The fat fuel molecule is referred to as a fatty acid, which is the simplest form of a fat molecule in the body. So in summary, the fuel sources for aerobic ATP production are carbohydrate, which is the glucose 6-phosphate from glucose or glycogen, as well as fatty acids. Now, the aerobic system is inefficient and efficient at the same time. It is inefficient because the rate at which ATP is produced is comparatively slow, and this is again related to the number of reactions that need to take place. So obviously, aerobic metabolism, whether the fuel is carbohydrate or fat, there are many steps involved. Now, the main reason why the aerobic system is inefficient, however, is because it is entirely dependent on oxygen availability. So as exercise intensity increases, there is more demand for aerobic ATP production, as you can see in this figure. This means there is more demand for oxygen availability in the muscle cell, particularly in the mitochondria. This means the aerobic system is highly dependent on other organ systems to get that oxygen to the muscle cell. Now, what organ system do you think this is? What organ system do you know becomes increasingly active as exercise intensity increases and ATP demand levels increase? Yes, it is the cardiovascular system. We all know as you increase intensity, heart rate increases proportionately. The heart also pumps more blood out per pump. Overall, there is more flow of oxygenated blood. The vascular system also redirects the oxygenated blood to the active muscles. This adjustment in the cardiovascular system is all for the sake of providing the oxygen to the muscle to meet the increasing oxygen demand of the muscle to support aerobic ATP production. So yes, the muscle is highly dependent on the cardiovascular system. And during our final lecture series of this course, we will talk about why training improves the aerobic system's ATP producing quote-unquote power at higher intensities. Meaning we will talk about why muscle cells are less reliant on anaerobic backup systems at higher intensities of exercise with training. And this involves enhancements in the cardiovascular system and its ability to provide more oxygen to the muscle to support aerobic ATP production. So more on that later. First, we need to discuss the basics. So those are the reasons why the aerobic system can be inefficient. But how is it efficient? The aerobic system is efficient because of the quantity of ATP produced per fuel molecule. We already talked about how anaerobic carbohydrate oxidation, aka glycolysis, is only partial oxidation, and you get only 2 to 3 ATPs worth of energy out of it. And we introduced the fact that after glycolysis, there can be further oxidation of that fuel molecule in the mitochondria via aerobic metabolism to get 30 more ATPs worth of energy out of it. For fatty acids, there is even more ATP produced upon aerobic oxidation. More on this in a few moments. Lastly, I must reiterate that aerobic ATP production is preferred, always active, and proportionate to the level of intensity or effort and ATP demand. Only at certain higher intensities, the aerobic system cannot meet the full ATP demand due to the insufficient oxygen availability. Again, this is called the anaerobic threshold. At this point, the anaerobic systems, as we already discussed, will kick in to fulfill the ATP demand that the aerobic system cannot. The anaerobic system does not, I repeat, does not just completely take over, and the aerobic system does not, I repeat again, does not simply stop producing ATP. So first, let's start off with carbohydrates, since we introduced the initial anaerobic portion of carbohydrate oxidation earlier. So the aerobic production of ATP from carbohydrate, or in other words, full carbohydrate oxidation, involves 3 processes. The first process, as we know, is glycolysis, which we also know is anaerobic. This process does not require oxygen, and it occurs outside of the mitochondria. So the full oxidation of carbohydrate involves initially an anaerobic process. During this process, the carbohydrate, more specifically the glucose 6-phosphate, is only partially oxidized and has only provided 2 to 3 ATPs worth of energy. Now, the next 2 steps are the aerobic processes that occur in the mitochondria. This includes the Krebs Cycle and the electron transport chain, or ETC for short. In order for aerobic production from the glucose 6-phosphate to occur, the glucose 6-phosphate first needs to undergo anaerobic metabolism via glycolysis. The reason being is because glycolysis is what provides the products that can enter aerobic metabolism in the mitochondria, and that product is pyruvate. So let's break this down even further. So as you see in this figure, we have glucose entering the muscle cell, and we know it converts to glucose 6-phosphate, which is the starting point. From here to the 2 pyruvates, this is glycolysis. And again, it is the anaerobic portion of carbohydrate oxidation. This is step 1, as indicated in the previous slide. You get 2 to 3 ATP out of it and some NADH, which we will discuss later. Now, as I said earlier, the pyruvates from glycolysis is the product that can be further oxidized in the mitochondria via aerobic metabolism. Of course, this would require sufficient oxygen. In that case, the pyruvates can be converted or oxidized to acetyl-CoA. And then aerobic metabolism starts from there with the Krebs cycle followed by the electron transport chain. I will demonstrate this in much more detail in a few moments. Now, as we discussed earlier during the talk on anaerobic metabolism, during situations where there is insufficient oxygen, like during post-anaerobic threshold exercise, an increasing portion of the glucose 6-phosphate, so not all the glucose 6-phosphate, but just an increasing portion of it, stops after glycolysis and ends in lactate, thus only being partially oxidized. This is described again as the lactic system or fast glycolysis. Now, to reemphasize, during post-anaerobic threshold exercise, the available glucose 6-phosphate goes in both pathways. Most of the glucose 6-phosphate is fully oxidized and continues past glycolysis into the aerobic metabolism, and some of the glucose 6-phosphate is partially oxidized and goes through this pathway ending in lactate. This is again described as fast glycolysis or the lactic system. So both full and partial carbohydrate oxidation occurs during post-anaerobic threshold exercise. Now, during pre-anaerobic threshold exercise, the majority, if not all, the glucose 6-phosphate is fully oxidized and thus follows this pathway following glycolysis into the mitochondria for further oxidation and ATP production via aerobic metabolism. Now, this information is very important for you to know for the exam. So if I ask the question, which pathway is active during post-anaerobic threshold exercise, the answer is both. Full oxidation via glycolysis followed by aerobic metabolism, as well as partial oxidation via fast glycolysis, which involves glycolysis and pyruvate simply ending in lactate. This is again described as fast glycolysis or simply the lactic system. So let's go ahead and take a look at the figure from the previous slide in much more detail using this figure right here that we used earlier to describe fast glycolysis. So again, this is the anaerobic portion of glucose 6-phosphate oxidation called glycolysis. So first, glucose 6-phosphate, as we know by now, is derived from two carbohydrate fuel sources, glucose or glycogen. And this is the starting point of glycolysis. Now, as it goes through glycolysis, it releases energy to form 2 to 3 ATP. Now, we also introduce these reactions right here. These are again called redox reactions, reduction oxidation reactions. So NAD oxidizes 3-phosphoglyceraldehyde and in turn reduces to NADH. Through this process, NAD steals electrons from 3-phosphoglyceraldehyde and is converted to NADH with the hydrogen containing a couple electrons. So from one glucose 6-phosphate, we get two redox reactions during glycolysis, providing two NADH, one right here and one right here. And again, these NADHs are electron carriers. Each of them carry a pair of electrons. Now, we will discuss soon what this NADH is actually used for. So we also get 2 to 3 ATP, as you see right here, and then we end up here with pyruvate. We got pyruvate here and pyruvate here, so two pyruvates. Now, before, I said that fast glycolysis or the lactic system is when the pyruvate is converted to lactate. And that is it for that glucose 6-phosphate. You just get 2 to 3 ATP and a couple of lactates. But for full oxidation, the pyruvate will continue to undergo oxidation by first being oxidized to acetyl-CoA. And this is the part that occurs in the mitochondria via aerobic metabolism. So since we have two pyruvates here, we end up with two acetyl-CoAs. So let's take a look at the big picture here and take a look at the three processes for full oxidation of a single glucose 6-phosphate. So in the blue box here, we have the first step, which again is glycolysis. And as shown in the previous slide, the glucose 6-phosphate is only partially oxidized, only providing 2 to 3 ATP at this point. And it also provides two NADHs through a couple redox reactions. And remember, the NADH carries pairs of electrons. This proton, as we discussed earlier, is also produced through the redox reaction, and we will discuss its role in a bit. So as we said, full oxidation of that glucose 6-phosphate involves pyruvate not being converted to lactate, but being further oxidized, and you can see that here. For each pyruvate, in this case there is 2, NAD will oxidize pyruvate, stealing a couple electrons from it. Again, this is a redox reaction. So pyruvate is oxidized and NAD is reduced to NADH, which again carries those electrons. Since we have 2 pyruvates here, we get 2 NADHs because 2 separate redox reactions have occurred. So pyruvate, when it is oxidized, it is converted to acetyl-CoA. And this is where aerobic metabolism begins with the Krebs cycle, which again occurs in the mitochondria. So generally speaking, the Krebs cycle involves a series of reactions which include redox reactions. For the sake of simplicity, I do not have any substrates, products, or enzymes listed here for the Krebs cycle. I will reserve that for your biochemistry course if you are planning on taking it. So just know that the primary purpose of the Krebs cycle is for those redox reactions. And through this, we fully oxidize the glucose 6-phosphate. Through those redox reactions, we get 6 more NADHs. We also have redox reactions that involve another oxidizing agent called flavin adenine dinucleotide, or FAD. And when FAD is reduced, it is reduced to FADH as it oxidizes molecules within the Krebs cycle. And the FADH generally serves the same purpose as NADH, which we will discuss in a few moments. Now through the Krebs cycle, some energy is released to form ATP. So you get 2 ATP out of the Krebs cycle from one single glucose 6-phosphate. But like I said, the main purpose of the Krebs cycle is not really ATP production, but redox reactions to provide electron carriers in the form of NADH as well as FADH. Now, after the Krebs cycle, the glucose 6-phosphate is again now completely oxidized, meaning the molecule is completely broken up and all the energy is released. So where did the energy go? Well, in glycolysis and in the Krebs cycle, the energy was released and used to form ATP. We get a couple ATP here and a couple ATP here. And the rest of the energy was transferred during the redox reactions, meaning the energy is now contained in the NADHs as well as the FADHs. So what happens to the glucose 6-phosphate molecule after full oxidation? Well, it doesn't just disappear. Things do not disappear. When you burn fuel, the fuel molecules don't just disappear. Mass is always conserved based on the law of conservation of mass. So let me explain. The mass of the glucose 6-phosphate is made up of carbons, hydrogens, and oxygens. And this is why carbohydrate is abbreviated CHO, carbons, hydrogens, and oxygens. So what happened to the carbons, hydrogens, and oxygens after glycolysis and Krebs cycle? Well, the hydrogens went to the NADHs as well as the FADH through those redox reactions, which again carry those electrons in the form of hydrogens. And the carbons and oxygens were converted to another molecule. What do you think that molecule is? The carbons and oxygens from the carbohydrate glucose 6-phosphate is now carbon dioxide, CO2. So for one glucose 6-phosphate at the end of glycolysis and Krebs cycle, it is converted to 4 carbon dioxides. As you can see, the mass of the glucose 6-phosphate is conserved but in a different molecule. Now, we all know where the CO2 goes. We exhaust it from our body through exhalation. This is the same concept as the fuel inside a car being oxidized or burned and converted to CO2, which is then exhausted out of the exhaust pipe of the car. So if someone asks, where does all the fuel in our body go after it is burned? We breathe it out. We also sweat and urinate it out as well, but in a different molecule, and we will talk about that in a second. All right, so let's summarize. Through glycolysis, starting with one single glucose 6-phosphate, you get 2 to 3 ATP and 2 NADHs, as well as 2 pyruvates. The 2 pyruvates are oxidized to 2 acetyl-CoAs, providing 2 more NADHs. As acetyl-CoA is further oxidized in the Krebs cycle, you get 6 more NADHs and 2 FADHs. You also get 2 more ATP. And at this point, the glucose 6-phosphate is fully oxidized. And the product of that is CO2, which exits our body via exhalation. All right, so now we have all these electron carriers in the form of NADH and FADH, and we still have much more ATP to produce. So where will the remaining ATP production occur? It will occur in the third and final step of full carbohydrate oxidation, which again is in the mitochondria and is aerobic in nature. This process is called the electron transport chain, or ETC for short. And as you can see, all the NADHs and FADHs go through the ETC. And through that, you get much more ATP production. So as you can see, there is a red box here around the ETC, which means we are going to dive into what the ETC actually is and how it works to produce ATP from these NADHs and FADHs in the next coming slides. So first we need to talk about the mitochondria, which as we know by now is where aerobic metabolism occurs. The Krebs cycle as well as the ETC occur in the mitochondria. So let's look inside the microanatomy of the mitochondria and look to see where the ETC actually occurs. So first, turn your attention to the inner membrane, which is the yellow border you see right here. As you can see, it separates the blue section from this section out here. The blue section is called the matrix, and the outer section is called the intermembrane space. So the inner membrane is what separates the matrix from the intermembrane space. And on this inner membrane is where the ETC occurs, particularly in these little folds called the cristae. So let's go ahead and zoom in on that inner membrane to see what's going on with the ETC. So here we have a very simplified visual and animation of the ETC. Here highlighted by this yellow wall is that inner membrane. And on this side of the inner membrane, you have the intermembrane space. And on this side, you have the matrix. Also, directly on the inner membrane wall, you have these components right here. These components are referred to as the ETC complexes. So you have complex 1 through 5. Now in complexes 1 through 4, there are specialized proteins called cytochromes. C-Y-T-O-C-H-R-O-M-E-S. Cytochromes. These cytochromes accept and transfers electrons down this chain of complexes. Also on these complexes, there are proton pumps that can pump protons across this inner membrane wall. More on this in a bit. Now, if you turn your attention to each side of this membrane, you see that there are protons on each side of the inner membrane, or wall, if you will. You have an equal number of protons on the matrix as the intermembrane space. In this example, you have 4 protons on each side of this inner membrane wall. Now, think of this inner membrane wall like a water dam that serves as a power plant. Energy is harnessed when there is a larger body of water on one side of the dam than the other. This creates a high to low gradient and potential energy. If you put a passageway in the wall, the water will flow from high to low gradient. So from where there is a larger body of water to the side that has the smaller body of water. And through the water rushing through this passageway in the water dam, you can harvest that potential energy by the rushing water turning a turbine. And in water dam power plants, we use this energy to produce electrical energy mostly. Now this is an analogy for the ETC. The protons are the body of water, and the inner membrane is the water dam. And what you are trying to create is a high to low proton gradient by putting more protons on one side of the wall versus the other. As more and more protons accumulate on one side of the wall, the gradient increases and the potential energy increases. At the end, there will be a passageway in the inner membrane for the protons on the high end to rush through. And this is where we will harvest that potential energy and use it to form ATP, which will contain that energy. So let's take a look at this entire process through this animation. So here we have the electron carriers that were produced in the first two steps of glycolysis from pyruvate to acetyl-CoA and from acetyl-CoA oxidation through the Krebs cycle. As you see here, the electron carrier is NADH, and on the hydrogen there are 2 electrons. As the NADH approaches the first complex in the ETC, the NADH is oxidized, meaning it will lose its electrons and thus convert back to NAD. And it will lose its electrons to the cytochrome on the first complex. Now, whenever the electron is transferred on the complex or to a complex, the proton pumps on the complex will be activated. So on the first complex, as the cytochrome accepts the electrons from the NADH, the proton pump in the first cytochrome will pump protons from the matrix to the intermembrane space across the water dam we call the inner membrane. Next, the cytochrome on complex 2 will accept the electrons from complex 1. And again, the proton pump on complex 2 will pump protons from the matrix to the intermembrane space. Now the body of quote-unquote water is getting larger on this side of the dam, and the potential energy is increasing because the proton gradient is getting larger and larger. And as the electrons get passed down the chain of complexes, more protons will get pumped from the matrix to the intermembrane space. And at this point, at complex 4, there is a large high to low proton gradient. Now, just like the water dam example, to harness this potential energy from this high to low gradient, we need a passageway through this inner membrane wall. And that passageway is represented by this 5th complex. However, keep in mind, this is not really a complex per se, but it is an enzyme called ATP synthase that works like a passageway with a literal turbine in it, just like the water dam. So at this point, the passageway can open up, and because of the high to low gradient, the protons will rush through this passageway called ATP synthase. And like I said, this passageway has a turbine mechanism. And as the protons rush through this passageway, the turbine will spin. And the energy from this will be used to rephosphorylate ADP to ATP, as you see right here. And that ATP will now contain that energy in that new phosphate bond. And this is how ATP is produced through the ETC. Now, FADH works the same way as NADH, but it enters the ETC a little further down the chain than NADH, and thus creates smaller proton gradients, and thus less energy is yielded. But that is not a critical point. The critical point is the process of the ETC and how it works to produce the ATP. So I would make sure you put this into full presentation mode and watch this animation repeatedly until you can understand what is happening with the ETC and how ATP is produced through the ETC. Now the next question is, what happens to those 2 electrons that were passed down the electron transport chain? This is where oxygen comes into play and why aerobic metabolism requires oxygen. Oxygen, as you see here by the letter O, is the final acceptor of those electrons. So for every 2 protons that come in through the ATP synthase, it combines with the 2 electrons. So if you have 1 proton and 1 electron, you have 1 hydrogen. So altogether, you get 2 hydrogens. And like I said, the oxygen will accept the electrons as they are within these hydrogens. So the oxygen and the 2 hydrogens combine to form what? H2O, a water molecule. So the final product of aerobic metabolism is H2O. So remember how I said the mass of the fuel molecule is conserved through its oxidation, while the carbons and hydrogens and oxygens of the carbohydrate fuel molecule, glucose 6-phosphate, was conserved, as I said earlier. The carbons and oxygens were conserved in the form of CO2, carbon dioxide, and the hydrogens were indirectly conserved in the form of H2O, a water molecule. And so the products of full carbohydrate oxidation are CO2 and H2O. And where do these go? Again, we exhaust them from our body. Carbon dioxide in the form of expiratory air, so exhalation as we breathe, and H2O through excretory fluids like urine and sweat. So again, when you metabolize or quote-unquote burn fuel, as demonstrated here, the components of the fuel molecule don't just disappear. It is conserved in the form of CO2 and H2O, and our body eventually expels it from the body. And then circle of life. The CO2 is exhaled, and plants use it to form oxygen, which we then inhale. The H2O is evaporated and will end up somewhere, and animals and plants will eventually consume it. Okay, so to summarize the total ATP from full carbohydrate oxidation, so oxidation of a single glucose 6-phosphate, through glycolysis, we get 2 to 3 ATP. I know it just says 2 here, but 2 to 3 ATP, depending on where glucose 6-phosphate was derived from. We also get 2 NADHs. The oxidation of pyruvate to acetyl-CoA yields 2 NADHs. And the Krebs cycle produces 2 ATP, 6 NADHs, and 2 FADHs. So the subtotal up to the Krebs cycle is 4 ATP, 10 NADHs, and 2 FADHs. Now in the ETC, each NADH is worth approximately 2.5 ATP. So you get 25 ATP from this 10 NADH through the ETC. And each FADH is about 1.5 ATP. So from these two FADHs, you get 3 ATP through the ETC. So in total, from one glucose 6-phosphate through full oxidation, you get 32 to 33 total ATP, which again is the full energy worth of a single glucose 6-phosphate. So remember, when glucose 6-phosphate only goes through fast glycolysis, or the lactic system, you only get that 2 to 3 ATP, and it ends with pyruvate converting to lactate instead of acetyl-CoA. So we know that lactate can eventually reconvert back to pyruvate, and then pyruvate can continue on into the aerobic metabolism, providing that 30 additional ATP. And this is why we said lactate has about 30 ATP's worth of energy still left. This is an important piece of info for the exam, and this is why I'm reiterating it. So please make sure you take careful note of that. All right, so in summary, the full oxidation of glucose 6-phosphate involves initially an anaerobic process called glycolysis, pyruvate not converting to lactate but oxidizing to acetyl-CoA, and acetyl-CoA undergoing further oxidation in the Krebs cycle. The NADH and FADH produced through the redox reactions in the first two steps, the glycolysis and Krebs cycle, go into the ETC where further ATP is produced. Also, please take careful note, as you see here, the ETC is where the majority of ATP is produced per fuel molecule. So like per glucose 6-phosphate. The ETC is where the majority of the ATP will be produced. So if I ask which of the following processes produces the most ATP per fuel molecule, the answer would be the ETC, electron transport chain. For glucose 6-phosphate, only 2 to 3 ATP from glycolysis and only 2 ATP from the Krebs cycle. You get 28 from the ETC. All right, so now what about the other fuel molecule that can provide ATP via aerobic metabolism? Remember, aerobic metabolism involves the oxidation of carbohydrate as well as fat. And I mentioned that the fat molecule used for fuel are fatty acids. This is the simplest form of a fat molecule, also known as a lipid molecule. So let's discuss the processes for the oxidation of fatty acids. Now, unlike carbohydrate, fatty acids are not involved in any anaerobic process, meaning fatty acids can only provide ATP via the aerobic system, which means fatty acid oxidation or metabolism only occurs in the mitochondria. And this is one of the reasons why carbohydrates, whether glycogen or glucose, is the most preferred and versatile fuel source for muscles, especially during exercise. I'll go ahead and repeat that. This is one of the reasons why carbohydrates, whether glycogen or glucose, is the most preferred and versatile fuel source for muscles, especially during exercise. This is the only fuel source that can provide ATP anaerobically and aerobically. So carbohydrates is a nice fuel source for both pre and post anaerobic threshold exercise conditions. This also means that your muscles always utilize carbohydrates as fuel, regardless of what type of exercise and intensity of exercise. So let's talk about fat oxidation. Now, as an important side note, when we say fat, we are talking about a lipid molecule like a fatty acid. We are not talking about tissue. We often use the term fat to describe adipose tissue because adipose is where much of the fat molecules in the body is stored. So many people simply call adipose as fat. So for the sake of not being confused, when I say fat, I am talking about fuel molecules, in this case, fatty acids. So fatty acid oxidation, like glucose 6-phosphate, involve three processes. The first process is called beta oxidation. As you can see, I crossed off glycolysis here because glycolysis is only relevant to carbohydrate oxidation. I may or may not ask a simple question as which of the following is not involved in the oxidation of a fatty acid. If one of the answer choices is glycolysis, then that would be the answer. Now the next two steps are exactly the same as for carbohydrate oxidation. We already know that the Krebs cycle and etc. are aerobic processes that occur in the mitochondria, but also know that beta oxidation is also an aerobic process that also occurs in the mitochondria. Remember, fatty acids can only provide ATP aerobically. So we know that the Krebs cycle starts with acetyl-CoA, which undergoes oxidation through the Krebs cycle, providing a few ATP, but mainly NADH and FADH, the electron carriers. So what do you think is the main purpose of beta oxidation? It obviously provides the Krebs cycle with the acetyl-CoA. This is somewhat similar to glycolysis, except there are different reactions involved, a different fuel source, and a different amount of acetyl-CoA provided by a single fuel molecule. So let's take a look at the entire process in a more visual perspective. So first, as I said, fatty acid oxidation occurs inside the mitochondria. There is no anaerobic process involved. So the first step is getting the fatty acid into the mitochondria, more specifically in the mitochondrial matrix, which is where beta oxidation occurs. Fatty acids, more specifically long-chain fatty acids, which we will describe in the next slide, needs an active transport system to get into the mitochondria, more specifically into the matrix of the mitochondria. So let's focus first on the left side here, outside of the mitochondria. Here we have a molecule called acyl-CoA, aka fatty acyl-CoA. This is not acetyl-CoA, but fatty acyl-CoA, which is different. And we call fatty acyl-CoA an activated fatty acid ready to be metabolized. So in general, you can view this acyl-CoA here in this figure as a fatty acid. So as you can see here, the fatty acid will combine with this molecule carnitine, producing fatty acyl-carnitine. This reaction is catalyzed by the enzyme called carnitine palmitoyl transferase 1, or CPT1, and you can see that in this legend right here. Now, fatty acyl-carnitine in this form can enter the mitochondria through the outer membrane. And to get into the matrix area through the inner membrane, this enzyme right here called translocase will provide that passage. So now the fatty acyl-carnitine is in the matrix, and it now needs to be dissociated back to fatty acyl-CoA and carnitine. And that is catalyzed by the enzyme CPT2, which does the exact opposite of CPT1. So now fatty acyl-CoA is dissociated from carnitine, and you see that right here. And the fatty acyl-CoA is now ready to enter beta oxidation. Again, this is just an activated fatty acid. And this entire transport system is referred to as the carnitine shuttle system. Now, as a side note, as indicated here in the third bullet, there are some evidence, albeit weak evidence, that may suggest that increasing carnitine bioavailability can increase the level of the carnitine shuttle system, meaning increasing fatty acid transport into the mitochondria. Now, many people in the supplement industry will say, well, this will enhance fat burn. This will enhance fatty acid utilization, and therefore may be a means to reduce body fat. However, the evidence shows that yes, increasing carnitine bioavailability through carnitine supplementation may support fatty acid transport into the mitochondrial matrix, but it does not result in enhanced fatty acid oxidation. And this is because fatty acid oxidation is on a per needs basis, meaning your cells aren't going to ramp up fatty acid oxidation just because. It will increase fatty acid oxidation when it needs it. And this is why carnitine supplementation is considered, at least in the scientific community, as one of the most ineffectual supplements out there, at least as it pertains to its claims, which is to increase fat utilization and fat quote unquote burn. It does not do that. So if you look at the weight loss supplement market, you will see a lot of supplements that contain carnitine and it's claimed to enhance fat burn. It does not do that. It's not evidence-based. But yes, it could help transport fatty acids into the mitochondria. Now, this may have some implications to performance, more specifically, long duration performance capacity. Now, we will talk more about that in my Bioenergetics course in KIN4540, so hang on tight for that when you take that course. Now, the fat molecule that undergoes oxidation for the purpose of releasing energy to produce ATP is called a fatty acid. And there are multiple types of fatty acids, primarily distinguished by the length of its carbon chain. So for example, as shown here, is the most common endogenous fatty acid, palmitic acid, which is a 16 carbon acid, or a 16 carbon fatty acid rather, meaning there are 16 carbons within this fatty acid chain. Now, I will only use this fatty acid as an example in exam questions or exam answer choices. So the ATP yield from fat oxidation will be relevant to palmitic acid. Just understand that the larger the fatty acid, the more ATP yield, and you will understand why as we go through this slide here. Now remember, as shown a couple slides prior, the first of the three steps for fatty acid oxidation is a step called beta oxidation. So the first step of fat oxidation or fatty acid oxidation is beta oxidation. And this again occurs only in the mitochondria. So to put it simply, beta oxidation is simply a process of breaking down the fatty acid into two carbon units. Okay, so pairs of carbons through each cycle of beta oxidation. And each pair of carbon equates to one acetyl-CoA. So through the complete beta oxidation of this 16 carbon fatty acid, you will get 8 acetyl-CoA entering the Krebs cycle, which is the second step of this 3-step fat oxidation process. Now remember, for glucose, there were only 2 acetyl-CoA provided to the Krebs cycle. So this should hint at the fact that you're going to get much more ATP out of this fatty acid than glucose. Now, other than acetyl-CoA, as you see here, we're going to have some more redox reactions during beta oxidation. This will provide NADH and FADH, which again are electron carriers that will be used in the electron transport chain, where again, the majority of ATP will be produced. So essentially, we have the same processes that come at the end of complete glucose oxidation or carbohydrate oxidation. So electron transport chain is the same. We'll get to that in a sec. So as alluded to earlier, you can see down here in the last bullet that this fatty acid will yield significantly greater ATP than glucose, 106 ATP versus the 32 to 33 ATP with glucose, or glucose 6-phosphate to be more specific. As you can see here in this figure of the fatty acid oxidation pathway, we have the first step here of beta oxidation. And in order for beta oxidation to be initiated, there is a couple ATP expended, but no big deal. And like I said in the previous slide, a 16 carbon fatty acid like palmitic acid, which is represented here, will provide 8-acetyl-CoA through complete beta oxidation from the 8 pairs of carbons. Now, beta oxidation will also yield through redox reactions 7 FADHs and 7 NADHs, which are again are electron carriers, which will be useful in the electron transport chain. More on that in a sec. Now, the acetyl-CoA here, the 8 of them, will enter the Krebs cycle, where again you'll have more redox reactions, providing more electron carriers in the form of NADH and FADH. And you will also see you get some ATP out of the Krebs cycle from these 8 acetyl-CoAs. It's going to be 8 ATP to be exact, not a whole lot, but you get some out of it. Also, if you see here, a large portion of the fatty acid is eventually converted to CO2, and we all know where that goes. All right, we expire it through breathing. So literally you are breathing out fat every time you use it as a fuel. So all these electron carriers, the NADHs and FADHs, enter the electron transport chain, which we went over earlier in detail. And again, this is where the majority of ATP will be produced. So if you add everything up, this is the total ATP breakdown from the 16-carbon fatty acid. And like I said earlier, I will only use a 16-carbon fatty acid example on the exam. So let's start up here. As I said earlier, there is initially 2 ATP expended before beta oxidation, and this is referred simply as fatty acid activation. Here, beta oxidation will provide 7 NADHs and 7 FADHs, your electron carriers. And here, the 8 acetyl-CoA will provide 8 ATP, 24 NADHs, and 8 FADHs via the Krebs cycle. And in the ETC, the 31 NADHs will give you about 77.5 ATP, and the 15 FADHs will provide about 22.5 ATP. So if we add all the ATP up from the oxidation of the 16-carbon fatty acid, there will be a net yield of 106 ATP, which I said earlier is significantly larger than what a single glucose phosphate would provide, which is 32 to 33 ATP. Now, this is generally why, speaking of energy yield, it is said that fat has more energy yield than carbohydrates. And this also explains why, when quantifying the energy of fat versus carbs, fats have greater caloric value than carbohydrates, calories being a quantification for energy. Now, just like glycogen being the endogenous storage of carbohydrates, there are also fat storage molecules stored in various cells in the body, especially adipose cells or adipocytes. These are the cells that make up adipose tissue. Now, they can be stored in muscle cells as well, but in relatively small amounts. The fat storage molecule is referred to as a triacyl glycerol, or more commonly triglyceride, or TAG for short. Now, each tag is made up of 3 fatty acids and a glycerol backbone, and that which holds it all together. Hence the name tri, referring to the 3 fatty acids, glyceride, referring to the glycerol. So let's continue to use the 16-carbon fatty acid as an example. So here we have one tag molecule with 3 16-carbon fatty acids held together by one glycerol backbone. Now, in order to utilize these fatty acids for ATP production via fatty acid oxidation, as we just discussed, the tag needs to be broken up, or in other words, catabolized. So fat catabolism is called lipolysis. Lysis referring to breakdown or catabolism of a large molecule, in this case a tag, and lipo referring to fat. The fat molecule here is the tag. So lipolysis is simply the breakdown of the tag, freeing up the fatty acids and thus becoming free fatty acids and the one glycerol. Now, the 3 fatty acids can undergo fatty acid oxidation and provide 106 ATP each, as we discussed, totaling 318 ATP. And the one glycerol can actually be converted into a glycolysis intermediate and thus providing about 19 ATP as an estimate. So this particular tag with the 3 16-carbon fatty acids and the one glycerol can provide about 337 total ATP, which is comparatively a large energy yield. So we can see that tags have a lot of energy and ATP producing potential, and this is why tags are known as our body's reserve fuel source. Tags are stored in high amounts, especially in our body's reserve fuel tank called adipose. And this is why we just refer to adipose as body fat, since fat, i.e. tags, are largely stored in this tissue. So when we overfuel our bodies, like when we consume more fuel or nutrients than we need, our bodies like to figure out a way to store up this extra fuel and therefore extra energy coming in. The body likes to store it up in the form of fat storage molecules like tags and then store it into the reserve fuel tank, adipose. Now, this is why adipose mass increases when we overfuel, or in other words, are in a caloric surplus. So seeing that tags are largely stored up in adipose, adipose is pretty much the distribution center for fatty acids to the rest of the body, such as muscle. So during situations in which there is a higher energy or ATP demand, such as during exercise, the adipose would distribute fatty acids to tissues or cells that need it, like contracting muscles. So the process begins by, again, catabolizing tags in adipose through lipolysis. This would free up the fatty acids, hence free fatty acids. The free fatty acids then enter blood circulation, where it can be mobilized to other tissues like muscle. Now, the muscles can uptake the free fatty acids and either store it up as a tag in the muscle cell, or like most cases, oxidize it for ATP production. So this is through the three processes that we discussed earlier: beta oxidation, Krebs cycle, and the electron transport chain. Now keep in mind, however, tags are also found in muscle, and muscle, when utilizing fat as a fuel, will largely tap into the localized fat stores in the muscle cell. But this is not to say that it doesn't utilize the free fatty acids distributed by adipose. Now, this whole process right here is referred to as fat mobilization, because fat molecules, i.e. fatty acids, are being mobilized from one area, like adipose, to another, like muscle. So now the question is, what fuels do our muscles utilize during exercise? Well, the answer is both. The better question is, which is the more preferred fuel source? The answer to that is, it depends. Now, based on what we know so far, many may think that fat would be the most preferred fuel by muscle, especially during exercise, because it has much more energy than carbohydrates. Remember, 106 ATP per a 16 carbon fatty acid versus 32 to 33 ATP from glucose. But here's the thing. It's not about how much ATP is provided. It is rather the rate at which ATP is provided. Okay, that is a point of emphasis. It is not about how much ATP is provided. It is rather the rate at which ATP is provided. In simpler terms, it's about the speed at which ATP is provided to the cell, like the muscle cell. Now remember, earlier in this lecture series, I mentioned that the rule of thumb is the more ATP yield, the longer it takes to provide that ATP. So what I'm saying is that fatty acids provide ATP at a slower rate than carbohydrates, like glucose or glycogen. This is about 2 to 3 times slower. So when the muscles start to contract, like during exercise, the rate of ATP demand increases, and thus the muscle likes to use a fuel that would provide ATP at a higher rate. This is carbohydrates. This is the glucose and the glycogens, more specifically the glucose 6-phosphate that comes from those two sources. So if you look here in this figure, in general, this is a depiction of the relative utilization of fat versus carbohydrates. Now, this is relative to the total energy expended. So at rest to low intensity exercise, when the rate of ATP demand is low, fat can be utilized as a fuel, since this is not a situation in which muscles need a high rate of ATP production. But as intensity increases, the ATP demand rate in active muscles increase, and thus fat becomes less of an efficient fuel source, because again, it provides ATP at a relatively slower rate than carbohydrates. So as intensity increases, carbohydrate utilization also increases. You also have to keep in mind that carbohydrates can provide ATP both anaerobically and aerobically. We discussed that in this lecture series. And we also know that fat can only provide ATP aerobically. So as you go from pre to post anaerobic threshold exercise workload, so from rest to low to moderate to high to max intensity exercise, you will need anaerobic ATP production to help back up your aerobic systems. Now, carbohydrates will be utilized at a higher rate during post anaerobic threshold exercise, so higher the intensity of exercise. This is why we consider muscle glycogen, your muscle's carbohydrate store, as the muscle's primary fuel source during exercise. And when glycogen runs out in the muscle, it is like a car with an empty tank. Exercise will be impeded despite the fact that there is still plenty of fat fuels available. When the glycogen, aka the carbohydrate store, is depleted in the muscle, exercise intensity will be difficult to maintain because, again, fat cannot provide ATP at the high rate that it is needed. So also keep in mind that this does not mean low intensity exercise is optimal for fat burning and therefore body fat loss. This is, again, a percentage of the total energy expended. So during rest to low intensity exercise, you're obviously not expending a lot of energy. So this high percentage is actually still a very low per gram amount of fat actually being utilized. So this specific topic of fat utilization and how that relates to fat loss, this is more of a topic in my exercise metabolism course. So we'll go into much more detail as you enter my 45-40 course. So just hang on tight. We will definitely get to that topic in the future. Okay, moving forward. Now let's go back and sort of summarize everything we talked about in relation to the aerobic anaerobic integration curve that we brought up in the beginning of this lecture series. So the white again represents aerobic ATP production, whereas the red represents anaerobic ATP production. So remember, as workload increases, your aerobic systems work harder and harder to meet the ATP demand. But it's going to get to this certain point of a certain workload where the ATP demand cannot be fully satisfied by the aerobic systems. And this is where your muscle cells need to depend on an oxygen independent system for ATP production, and that is called anaerobic metabolism. That is again represented by this red. So you can see the anaerobic system is simply a backup for the aerobic system. So we sort of compartmentalize this curve into three sections. So we can see at various workloads or at increasing workloads what is happening in terms of aerobic and anaerobic metabolism. So if you look down here, this is like rest to low intensity exercise. The aerobic system is efficient in meeting the ATP demand. Why? Because the ATP demand rate is pretty low. There's sufficient oxygen availability because the oxygen demand is not very high. So this is where aerobic metabolism can efficiently produce ATP to meet the relatively low ATP demand of the muscle. There is no acute fatigue, and exercise can be performed at steady state for relatively long duration until you obviously just run out of fuel, which is going to be hours if you have a full fuel tank. So as you increase intensity, say within this zone right here, your aerobic systems start to ramp up to meet the increasing rate of ATP demand by the muscles, because your muscles are obviously contracting harder and needs ATP at a faster rate. So the cardiovascular system, which we'll talk about towards the end of the semester, is working harder to deliver more oxygen to support the aerobic metabolism, right, in the mitochondria. The aerobic ATP production increases with effort or workload and ATP demand. As we discussed in the previous slide, is becoming less reliant on fat and more reliant on carbohydrates because, again, carbohydrates can provide ATP at a faster rate than fat. So during exercise, and this is sort of representative of most exercise that we do that we consider like cardio or just some kind of steady state exercise, like running on a treadmill or something like that. Again, we have increasing rate of aerobic ATP production. And then we get to this point right here, a workload past this point. Remember, this point right here where the aerobic system cannot fully meet the ATP demand, and therefore you need your anaerobic backup systems to meet the full ATP demand. This point right here is called your anaerobic threshold. This is post-anaerobic threshold exercise. This is pre-anaerobic threshold exercise. So during post-anaerobic threshold exercise, aerobic ATP production is insufficient to meet the full ATP demand. It can't cover the full ATP demand. And this is because there is insufficient oxygen availability. Okay, it doesn't mean there is no oxygen. It just means there's not enough oxygen available for the aerobic system to produce ATP to the point where it can meet the full demand. And this is why the muscle cell relies on a oxygen independent system called anaerobic metabolism. So anaerobic ATP production compensates for this. It fills the gap here so that the muscle cells can fully meet the ATP demand. So what is happening during post-anaerobic threshold exercise? Again, aerobic metabolism is at its near capacity and cannot meet the ATP needs of the muscle cell. So we rely on fast energy systems that are anaerobic, that does not require oxygen for ATP production. So fast ATP producing systems. We have the fast glycolysis system or the lactic system where you get 2 to 3 ATP from a glucose or glucose 6-phosphate rather, really fast, and ends with lactate, which again, I said is primarily reutilized as a fuel source when the conditions permit, or it can be also used to be recycled back to glucose in the liver through gluconeogenesis. And the other anaerobic system is the fastest ATP producing system, one reaction catalyzed by creatine kinase, and that is the phosphagen system or the alactic system. So these two anaerobic systems are represented by this red curve here, and it just backs up the mitochondria, the aerobic systems. Now, post-anaerobic threshold, as we know, is fatigue inducing. This is a state where muscles are inhibiting itself. And the muscles become inhibited because post-anaerobic threshold exercise is stressful for the muscle. Now, like I said in the last slide, there is much more details related to bioenergetics in my 4540 class, my exercise metab course. And one of the major topics is muscular fatigue. Okay, so this is sort of laying the foundation for the more advanced topics that we're going to discuss as it relates to muscle fatigue in your subsequent course. Okay, so more on that later. Now, going back to what we discussed before in terms of fuel utilization during exercise, especially in response to increasing exercise intensity, this slide here expounds a little bit further. So again, in relation to the 3 sections on this aerobic anaerobic integration curve, in this section right here, again, this is where aerobic systems are efficient to meet the ATP demand because, again, the ATP demand is very low. More specifically, the rate of ATP demand is very slow. So the primary energy substrates or the primary fuel source here can be adipose, liver, and muscle lipids or fats. Muscle and liver glycogen, you can utilize those as well, and also blood glucose. So you can utilize all these different fuel sources. As you increase intensity into this zone right here, still pre-anaerobic threshold, but increasing exercise intensity, as we discussed before, there's sort of a little bit of a shift in the preferred fuel or energy substrate. So muscle glycogen, so this is localized carbohydrate stores, already stored within the muscles. This is one of the preferred fuel sources as you increase your workload. So increase the activity of the muscle and the rate of ATP demand. We can also utilize lactate if available. Like I said, lactate can be used as a fuel source. This is just a partially broken down carbohydrate. And we become less reliant on fats or lipids as effort increases. Again, it's about the rate at which ATP can be provided, not just the quantity of ATP provided. So fats can provide a lot more ATP, but it's going to provide it 2 to 5 times slower than carb sources like glycogen. So as we get into post-anaerobic threshold, high to max workload exercise, of course, your primary fuel substrates or energy substrates rather starts to shift. Becomes highly reliant on muscle glycogen, blood glucose, phosphocreatine, and very low reliance on fats. Again, this post-anaerobic threshold exercise scenario is characterized by an extreme or very high rate of ATP demand. So it makes sense that the muscle cell relies on fuels that can provide ATP at a very high rate. Okay, and so those are listed here. So you got your carbohydrate stores, your glycogen, Another carbohydrate source, blood glucose. You have your phosphocreatine for the phosphagen system. And of course, again, fats or fatty acids are not the most reliable fuel when ATP demand rate is highest. Okay, and finally we come to the last slide. This is a summary of aerobic metabolism as it relates to exercise and meeting the ATP demand aerobically in skeletal muscle during exercise. So make sure you go over each of these bullet points as a summary of everything we discussed in relation to aerobic metabolism.